then I will speed up a bit so that I don't. So next is on the shotgun metagenome profiling. So so we know that, for shotgun metagenome profiling, our goal is to actually identify the species that are present in a sample. For example, a soil sample or a fecal sample, and also we want to know the relative abundances as well as look into, some of the functional information or gene information in that particular sample itself. So hi-fi reads, there have been, many of the publications which I have shown you in the one of the slides earlier as well, where, a lot of researchers are actually using our hi-fi for hi-fi metagenome profiling because we can actually assign taxonomy with high precision and recall. And each hi-fi reads, because we are, so long, right? You can actually have, multiple genes intact per read, which can actually provide you richer information about the genes that occur in single microbe and maybe co-express as well. So 80 or to 90% of the hi-fi reads will have at least one functional annotation, where I can show you in one of these case study by Soltea Therapeutics. So they were actually doing a comparison between they were running these fecal microbiome samples on both short read and long read sequencing. And they found that, only 34% of the short reads can be annotated. I mean, it's logic, right? Because, short read is only 100 to 150 base pairs, right? So uh, genes are usually, longer compared to 100 to 150 base pairs. So that's why, only 34% of them can be annotated. Whereas for hi-fi reads, right, up to 70% of our hi-fi reads can be annotated. And oops. Yeah. And each hi-fi reads actually has an average of four annotations where short reads have at most one. So you need actually 10 times fewer hi-fi reads to generate the same number of functional insight compared to what you're doing on the short read sequencing. Okay. Then next is on the last applications that I would like to just further elaborate is on shotgun metagenome assembly. So for the short metagenome assembly, so just now metagenome profiling is we want to actually profile to see, in a microbiome community, how many, uh, what species are in that community itself, and also, what are the relative abundance and also the gene function or the functional information, right? Whereas for metagenome assembly, we will usually sequence deeper so that we want to actually, assemble a representative genome from this microbiome sample. So instead of, doing, pure culture and doing microbial whole genome sequencing for a single culture, for metagenome assembly, we are actually trying to assemble the genome in a microbiome sample itself. Okay, but there are certain challenges, of course, right? Because there are multiple species that are present, and there's uneven and unknown coverages, and of course, there are things like difficult regions as well in these microbe genomes. So if you compare between the short read metagenomics and if you compare to what we are doing on hi-fi itself, right? So as I mentioned in earlier as well in the microbial whole genome sequencing application. So because our reads are are long and accurate itself, right? So once we sequence through the microbiome sample itself, we are able to generate reads that are very long, and some of them, will actually, already form like a closed genome itself. So by doing that, we can actually, form complete genomes and genomes that have lesser gaps compared to what you have on the short read side. So for the short read side, you rarely will be able to get a whole genome of a bacteria based on 100 to 150 base pairs, right? You will still need to use bioinformatics tools to actually assemble them to get the full genome itself. But a lot of time, because their reads are so short and then there are a lot of complex regions, you'll not be able to get a full genome, and there will be a lot of gaps in the genome itself. And of course, compared to another long read technology itself, right? So we know that metagenomes offer contain many closely related strains, And some of the closely related strains only have a difference of only about like 3%. 3% is a common cutoff for defining a distinct species. So if the difference between the closely related strands is less than the error rate itself, maybe less than 3%, the error correction can actually erase these real differences. And then you might miss those strands itself. You might call them as the same species and same strand because of the error correction that has, corrected the error away. Whereas, for hi-fi reads, because we are 99.9% accurate already, you do not need any error correction. And we can actually be able to actually assemble uh, the genomes properly and to be able to resolve strain level information and have a complete genome itself. Yeah. So just to show you a paper that, compare between the nanopore technology as well as the PacBio hi-fi technology with and without short read polishing. So if you look at the darker green or the darker colors, right? So darker green, darker yellow, darker purple. So these are all the uh, sequences that are just based on the pure long read sequencing itself. Whereas the lighter ones are the after the researcher polish it with the Illumina data. So you can see that, for PacBio data itself, right, with or without polishing, we are already, our ideal score is so much more higher compared to what ONT can actually offer. And some of them, you can see that, the score actually dropped down with the polishing itself. So this is the reason why we are saying, we actually don't need short read polishing for most of our whole genome sequencing as well as the shotgun metagenome assembly pipeline. Yeah, and lastly, just to show some of the recent preprint as well, comparing the different technologies and, for our PacBio data, we actually yield up to 70 times more complete metagenome assembled genome or max compared to Illumina or 2.5 times more than ONT itself. And our PacBio outperforms ONT in all the key metrics, for example, read quality, mean median read length, and 50 read length. And our max usually have higher completeness and lower contamination. And in terms of cost as well, because of all the different, prep workflow improvement and platform improvement that I mentioned earlier on as well. So the researchers actually, calculated that the cost of running PacBio is actually the most cost effective. And another comparison to show you, comparing hi-fi reads versus short reads in one of the paper looking at soil microbiome. So you can see that, in this particular paper, they generated around 629 gigs of hi-fi data, whereas they generated around 3,400 gigs of Illumina data. And you can see that the number of max that they actually are able to actually achieve in the particular sequencing run. So you can see that for PacBio data, they are able to generate up to a 2041 max, which 587 is a single contig high quality max, and 315 is high quality max, and 1,000 of them are medium quality. So this is, you can see that is the differences between the data that you can get from hi-fi reads and short reads are so significant. So for Illumina, they are only able to get around 307 max in this particular study itself. So if we calculate, the consumable costs and we divide by the number of max itself, you can see that, for each of the max that we actually achieve, we can actually, it's only cost $5 on PacBio. And for high quality Macs, it costs around $11 on PacBio, which is, such a big difference compared to what it is to run on the short read platform. Okay, so this is my final slide just to show you all, we do have, all the different workflows and different protocol for, for full length 16S, KeyNext 16S, as well as if let's say you're looking at fungal or eukaryote amplicon profiling, we do have application node to help you all to do that. And then for shotgun metagenomics as well, we do have our PacBio whole genome sequencing library prep workflow that you can actually follow uh, to actually, perform these experiments. Okay, so with that, I think that's the last of my slide, and I will pass it on to Ting Ting to go through some of the tools that we have for microbial and microbiome research from PacBio.
Thank you, right.
Mm,
Maybe you please share the screen again,
oh,
because we
yeah.
really want to discuss about the wet lab, and then
Oh,
we
sure.
move on to the bioinformatics later.